Hi students, now discuss about cell-mediated immunity. It is the second type of the immunity. Based on their response against the microorganisms, antigens in the body, immunity is two types. Student, in this first one is humoral immunity. Second one is cell-mediated immunity. Now what about the mechanism? Now discuss about what about the mechanism involved in the cell-mediated immunity. Student, in this type of the immunity by Ctls, that means cytotoxin T lymphocytes, and NK cells, natural killer cells. This is the amount of the immunity, cell-mediated immunity is by cytotoxic T lymphocytes and the natural killer cells. In this, it is effective against exogenous and endogenous antigens. Student, okay, it is the cell-mediated immunity shows action against the effective action shows against the exogenous antigens and the endogenous antigens. Okay, exogenous antigens, the type of the antigens generated outside of the cell, exogenous antigens. Endogenous antigens generates in inner part of the cell, okay, cytoplasmic region of the cell, that is endogenous antigens. Both type of the antigens are, these are the particles of the microorganisms, pathogens. Next, in this type of the immunity, if you observe, entire the cell mediated immunity is with do not production of antibodies. Antibodies are not produced in cell mediated immunity, but antibodies are produced in humoral immunity. This is the difference. Next, APCs, antigen presenting cells. Which are the antigen presenting cells? B lymphocytes, macrophages, dendritic cells. These are the antigen presenting cells. Antigen presenting cell process exogenous antigens, student, and altered self cell process endogenous antigens. Next, antigenic fragments, antigenic determinants, okay, antigenic parts, antigenic fragments display on the membrane of APCs, altered self cells, recognized by T helper cells and TC cells. T lymphocytes, that means T helper cells, TC cells. T helper and TC cells, these are recognizes antigenic fragments which are displays on the membrane of the APCs and altered self cells, student. Next, T cells binds with antigen presenting cells and altered self cells, you know, T helper cells and TC cells. These are both type of the T lymphocytes, binds with antigen presenting cells and altered self cells, causes the production of the activated T helper cells and the memory T cells, student. And activated T helper cells, when T helper cells are activated, when these are binds with the antigen presenting cell and antigen presenting cells, okay, student. At the same time, T helper cells, activated T helper cells produces interleukins. Interleukins are soluble mediatory proteins, small molecules, okay. These are the interleukins produced in plasma of the blood. Interleukins are one type of the cytokines produces from T helper cells. It is important. And transform activated TC cells Activated TC cells. Interleukins are produced from the T helper cells and changes into activated, acts on the activated TC cells into cytotoxic T lymphocytes. These are also called killer cells, student. And CTLs. What is the function of the CTLs? Okay. Cytotoxic T lymphocytes attaches to the infected cells in the body, virus infected cells in the body. Okay. Infected cells, and releases two types of the enzymes. One is the perforin, another one is the granzymes. Both type of the enzymes, perforins, granzymes, releases by CTLs, cytotoxic T lymphocytes, student. Okay. In this, student, if you observe the mechanism of the cell mediated immunity, this is the antigen presenting cell. Okay, this is the exogenous antigen, student. Exogenous antigens enters into the inner part of the antigen presenting cell, for example macrophage or dendritic cell, B cell. Okay, and this is the antigen processed into smaller particles which are called antigenic fragments. Okay, this is the antigenic fragment is present to the T helper cells. Okay, presented through the class 2 MSC molecule, student, present on the surface of antigen presenting cell. Antigen presenting cell, if you observe cell membrane of the antigen presenting cell, that consists a class 2 MSC molecule. By the help of the class 2 MSC molecule, these are the antigen fragment is presented to the T helper cell. T helper cell that is recognized by the TCR, T cell receptor. It is the T cell receptor, student. It is the CD4 molecule present on surface of T helper cell. Okay, this is the antigenic determinant is recognized by the T helper cell. T helper cell is binds with the APC. Okay, next, what about endogenous antigens? Alter self cells, student. Endogenous antigens are processed and converted into antigenic fragments. This is the altered self cells. Altered self cell, all nucleated cells in the body called altered self cells. Infected cells in the body, altered self cells. Okay, antigenic fragments are presented through the class 1 MSC molecules of altered self cell. This is the antigenic fragment which is presented by altered self cell, that is recognized by TC cell. TC cells binds with the altered self cell, student, and student, T helper cell is activated. T helper cell is activated and produces interleukins, secretes interleukins into the plasma of the blood. These are the interleukins stimulates the TC cell into two types of the cells, student. One is the memory TC cell and the second one is effector cytotoxic T lymphocytes. TC cell is changes into two types of the cells, memory TC cell and effector cytotoxic T lymphocytes. These are the cytotoxic T lymphocytes are connected with the infected cells in the body. Effector Ctl, that is binds with the infected cells in the body, which are the virus infected cells in the body, which are the pathogen infected cells in the body. These are the cells are bind with the cytotoxic T lymphocytes are the killer cells. After binding, this is the killer cell CTL, cytotoxic T lymphocyte, releases two types of the enzymes Which are the two types of the enzymes? One is the perforin, second one is the granzymes. Student, these are the perforins. These are the perforins releases from Ctl, and students, these are the forms pores on the cell membrane of the infected cell. Student, these are the pores. These are the pores are formed by perforins. Okay, after the formation of the pores on the cell membrane of the infected cell, and granzymes are entered into the inner part. Student, these are the granzymes releases from the Ctl. These are the granzymes. Okay, these are the granzymes by the help of the pore-like arrangements which is formed by perforins, and granzymes enters into the inner part. Of cytoplasm of the infected cells. Student, okay, granzymes enter through pores, activate cospases. What about the cospases? Cospases are these are the enzymes. Cospases are the enzymes, and this is the protease enzyme. Caspase is the protease enzyme, and helps in the breakdown of the proteins which are present in the infected cell. Okay, and finally this is the caspase causes the programmed cell death. Okay, entered by the help of the protein breakdown. Proteins are break down into simple polypeptides, small polypeptides, or single amino acids by the caspase enzyme. It is the protease enzyme. Student, okay, and finally it leads to the destruction of the programmed cell death. Destruction of the cell or programmed cell death. Okay, student, helps in destruction of the infected cell, that is also called apoptosis. Student, okay, finally it is leads to destruction of the cell, that is called apoptosis. Apoptosis means a dying of the infected cell. Okay, it is about the cell mediated immunity.